Hello, I'm Allison Michael. I'm a scientist with the New Hampshire Academy of Science. I have done doctoral research in materials chemistry, but my undergraduate degree is in biochemistry. Uh, I work with Kelly Salmon, to whom you can direct any questions throughout the presentation. Um, she'll either answer them right away or interrupt me, or you can ask them at the end. And that's Kelly chatting, saying.
Yes, sorry, that was me.
No worries. So this is a collaboration between the New Hampshire Academy of Science and the Claremont Makerspace. We're also joined by Catherine Dwan, one of our student assistant scientists, who's been helping us particularly with our protein modeling projects this summer. So let's get started. So tonight we're going to cover what proteins are and where they come from, what this all has to do with COVID-19 as advertised, where you can go to learn some more information about proteins and also get your hands not dirty because it's on a computer, but some hands-on interaction with relevant proteins to the current public health situation, and then how you can sign up your computer to help curtail COVID-19. So starting out, what are proteins and where do they come from? So proteins are complex macromolecules, which means they are really big molecules and they have lots of different features and lots of different layers of structure, and we're going to get into that a little bit tonight. But just an initial point of view, we can think of it as amino acids strung together like beads on a string. So you can see this here, this nice little necklace. Every different colored ball is an amino acid. And in this case, the string or the polypeptide backbone is made of peptide bonds, which are covalent bonds that form between carboxylic acid and amino groups. So here you can see kind of an archetypal amino acid structure. We have the carboxylic acid group on the right highlighted in yellow, and the amino group on the left highlighted in red. And so it's this covalent bond that forms between these two groups, strings them together. So it bonds with its neighbor. And then we have this side chain here, it's called R. We just use that in chemistry as a variable, meaning it can be any one of a number of things. We'll get into more detail on the different side chains and their structural and chemical differences in a little bit more detail later on. So proteins have a lot of different functions within your body and other biological systems. They can be antibodies and bind to antigens like we discussed in previous lectures in this series. They can act as enzymes, so they can speed up and facilitate biological reactions that play a role in metabolism. They can act as messengers like hormones. They go from one part of your body to the other to say what's going on in the system, how things are doing. They can be structural proteins like keratin and collagen that really hold us together and make the fibers of our being. They can also be transport proteins. So we can think about this in ways that they carry parts and important things from one part of your body to another. And this actually right here is a transport protein. So this is hemoglobin, which is the oxygen transport protein that's in your red blood cells. So it has a heme group right there. You can see it a little bit in gray with an iron in the middle. And this is what ferries oxygen to all your different body systems. So I said these were complex molecules, so they have all sorts of different layers of structure. So we're gonna start at the bottom and build our way up. So we're gonna talk about their primary structure. So when we think about primary structure, it's the order of the beads on the string. So the little colored pattern you saw, that's the primary structure. In more sciencey terms, it's the sequence of the amino acids. So here we have our archetypal amino acid here, and what's going to change from amino acid to amino acid is the side chain. We call them amino acids because we have this amino group and also this carboxylic acid group. So amino and acid. Sometimes biochemists get creative with their naming. This is not one of those times. So there are 20 amino acids that are used in the human body. In this figure, they're color-coded by their chemical properties. So some are hydrophilic and really interact a lot in a water-rich environment and have all sorts of complex binding, hydrogen bonding going on. Some are completely nonpolar. They like a really oily environment. They really enjoy being in the fatty areas of a lipid bilayer in your cell membrane. And some have hydroxyl groups, so they can kind of, you know, do a bunch of different things depending on what's going on around them and everything like that. So these with solid colored circles are amino acids that your body can make itself. And the ones in dashed circles are called the essential amino acids, and you have to obtain those from eating food. So different fruits and veggies, some meats, all have these essential amino acids that your body still needs to make the proteins that you need to live, but your body can't make them itself. So this is primary structure. It's the sequence. So moving on to secondary structure. Secondary structure is the localized conformation of the polypeptide backbone. So it means really local, not quite on the what exactly are the side chains level, but on the what are the neighbors doing, what are people around the corner doing sort of thing. It's how that backbone twists and folds. And this is where we begin to look at protein folding, which is really what gives the proteins their structure and therefore their function. So there's two really key typical forms of secondary structure, which is what we're going to go over now. There are some other kinds of secondary structure that are really just not that common. So here we have alpha helices on the left in both of these figures. So here and here. And so these form a right-handed helix, and we see that there's interactions between the carboxylic acid group and the amino group of these amino acids that cause it to fold up into this helical structure. It's a right-handed helix, so it goes around like this as opposed to a left-handed one. And then we have a beta pleated sheet where we see these intermolecular bonds, these hydrogen bonds forming between the chain, same chain still. This is all the same protein in this order right here, leading to a more folded structure. So this is more what it looks like in person. These are the cartoons that we use to describe them. And Catherine will show you where it really helps to know what these cartoons mean when you start to play around with the structures themselves. So alpha helices, there's not much change. With the beta pleated sheets, we just make a really flat arrow that sometimes has a little bit of twist to it. So secondary structure, it's locally what's going on in the neighborhood. And then tertiary structure is the overall shape of the folded polypeptide chain. So we look at it and we see this noodly thing over here and we're like, yes, that's the protein, that's its shape, that's going to inform what it does. You can see there's some pleated sheets and some helices drawn in here that help to create this tertiary structure. So the tertiary structure depends on the secondary structure, which depends on the primary structure. So it's layers on layers on layers. Proteins are like onions and ogres, they have layers. And what helps to fasten and stabilize this tertiary structure are a variety of intermolecular.
Excuse me, Allison. I hope everybody appreciated the Shrek reference.
Thanks for pointing that out.
So we stabilize this tertiary structure with interactions that can happen between the side chains. So we can see some ionic bonds forming between charged amino acid side chains. We can see hydrophobic interactions happening between those oily amino acids that really want to sequester away from an aqueous environment. So we think of our cells as being a really watery place to be. Our bodies are made up of mostly water, and that's not outside the cells in a separate bag, that's inside the cells. So we're thinking of all of this happening in a watery environment. So just like the olive oil in your vinaigrette dressing will sequester itself away from the vinegar and the water, so too will these side chains sequester away from the watery environment. We can also have disulfide linkages. So it's between two sulfurs in amino acid chains. So these are called cysteines, and they can form a disulfide linkage. This is actually what gives curly hair its curl, is disulfide bonds in the proteins. And then we can see hydrogen bonds. We talked about that a little bit earlier, where hydrogen is attracted to the electrons on a more electronegative atom. Electronegativity just has to do with these atoms prefer to have more electrons around them. And so hydrogen isn't very electronegative, but it's more positively charged, and it sees that negative zone and it's attracted to it. So all of these help to stabilize this tertiary structure. And then there's also quaternary structure, which we're not going to talk about. It gets really complicated. It has to do when you have multiple polypeptide chains interacting with one another. So these folded chains then get used like Legos and stuck together. And often we see those interactions occurring where a lever gets switched on one side, and that causes the tertiary structure to change of one. We call it then a subunit. It's part of a bigger protein. So the one chain, the structure changes, and that causes something else to happen in a neighboring subunit. So quaternary structure is we just take tertiary structures and stick them together. Okay, so that's a whole lot going on. So those are all the layers of protein structure that we want to think about and that inform our understanding of the way proteins behave and the jobs that they do and where they end up being in a cell. So that's proteins and what they are. Where do they come from? So this is called the central dogma of molecular biology, and it's really kind of just how life works in general and our understanding of that. So as most things in biology come from, it comes from DNA. So we have this cute little chromosome over here, and so this chromosome is made up of that DNA double helix that's wound even tighter and roped around proteins called histones that give it this really tight package structure. So you have enzymes in your body that will help to unwind that and stretch it out into its double helix form, and then it'll pull it apart and what's called transcribe it. So we're going to take it from DNA, which is deoxyribonucleic acid, into RNA, which is ribonucleic acid. So the backbone's a little bit different, not a whole lot, so the letters are pretty similar. So it's going to see this code in the DNA, and then it's going to add complementary base pairs. So A's binds with T's, or T's bind with A's, and then A's would bind with a T if it was DNA, but in this case, it's going to bind with a U, that's uracil, because, you know, RNA just has to be a little bit different. So we see this intrinsic coding is slightly different, and then we have an M in front of this RNA. RNA has lots of different jobs in the cell. We're going to focus on proteins and the central dogma tonight. So transcribing is taking the DNA and turning it into RNA, and then we're going to translate it from our little nucleotide realm of DNA and RNA into a protein. So we send this messenger RNA, we're going to package it up, send it outside of the cell nucleus where the DNA lives, and to a ribosome, which is going to look at these 3-letter codons. So it groups them in groups of 3. And so that 3-letter code tells the ribosome, oh yes, I want to put a methionine there. And then I'll attach it to an isoleucine, and then to a serine. So it goes 3-letter code by 3-letter code by 3-letter code, and then there's another 3-letter code that says stop. That's the end. So throughout this whole process, there's a lot of other different stuff that goes on. Some stuff can happen in when the mRNA gets packaged, it can cause splices. And so what is exactly transcribed isn't exactly then what gets translated. And then once it's translated, there can be helper proteins that help it to fold in a certain way or cleave off other bits of it. But in general, we go from DNA to mRNA to a polypeptide, and a polypeptide is just a protein. In the same way that a polymer is a plastic. So it's lots of little different units strung together by covalent bonds. Okay, so now that everyone is having flashbacks to their high school and college biology and biochemistry classes, you're probably wondering, what does all of this have to do with COVID-19? So COVID-19, as we all know, is the disease caused by the SARS-CoV-2 virus. And so SARS-CoV-2 is a coronavirus. It has this crown of spike proteins around it. These little bright red triangle guys, they're triangles because they're a trimer. So they have quaternary structure. So it has 3 protein subunits all together interacting. So they're stuck into this membrane of this virus. And they are, because they stick out and are really obvious, they're one of the key targets for therapeutics for COVID-19, and they play a role in the entry of the virus into your cells. Specifically, they enter your cells by binding with the ACE2 receptor, which is Peter's favorite receptor, I think, right now. It plays a role in the angiotensin II system, and it's great and awesome and does a lot to regulate your blood pressure. But for here, right now, it's been hijacked by this virus to get into your cells because it regulates blood pressure. It's present in a lot of tissues. And that's why we see wide-ranging systemic effects when we have an infection with SARS-CoV-2. So there's a fusion and a pre-fusion conformation, but it's this binding with the ACE2 receptor that allows the viral entry. Peter, do you want to talk about the pathway more, or is that enough background? Okay.
No, it'll bore people.
Okay.
And the only thing I would say is that it has a lot to do with really why people are susceptible probably in different, like one of the really big risks for COVID is obesity. And it turns out that obesity has a lot of things related to the ACE receptors that ramp up, and that there's also interleukin 6 and interleukin 8 that are ramped up in obese people. So why do obese people, it's probably the biggest risk group I think that's going to turn out to be. And they just have a lot of things going for them. And adipocytes release, when you have an overabundance of large fat cells, they release inflammatory products. So I think one of the things that's going to happen probably when they give some of these vaccines, they might pre-treat some obese people with certain anti-inflammatories because they're ACE2 receptors and all these other things. It's really complicated. I will put it into perspective though. When I did my physiology in my PhD, which most of my PhD was in physiology, although it was hematology, we didn't even know about the ACE2 receptor. We knew just about renin-angiotensin that raises blood pressure. And we knew about other things that lowered blood pressure, but we didn't know about this ACE2, which is the opposite. It lowers your blood pressure. So if you interrupt that system, you're going to mess around with high blood pressure. And another thing that obese people have is high blood pressure. So we put all those things together, and it does kind of give you the 10,000-foot view of why this whole thing is so important.
So that's 10,000 feet. Zooming in about as close as we can zoom into this.
There you go.
You can see there's this alpha helix right here, and the I have these labels wrong. Okay, ignore this. This is the ACE2 receptor. This is the spike protein. So this is the internal helix of the ACE2 receptor. We see this nicely made alpha helix right here. And then this is the spike protein and its receptor binding domain, which is the part of the protein that binds with the receptor, is right here. In particular, we look at this loop right here as really interacting with the end of this helix right there. So you're like, wow, we talked about helices and we talked about beta sheets, and I can see there's beta sheets there and helices there, but what are these skinny noodles? And the name for the skinny noodles is random coil. And it just means we can flop around and do whatever. There's not a whole lot of interactions holding that in place. So that's the spike.
So the spike protein has a lot of loop-de-doos in it for the size of that molecule, at least that part.
Yeah, so this is just a portion of the spike protein. This is just the part that plays a role in receptor binding.
Yeah.
It's actually, there's 3 really big things and then a part that lodges it in the membrane as well. So,
okay.
Yeah. So now.
That's kind of the head of the spike protein?
Yeah, this is the head of the spike protein, the specific part that interacts with this part of the ACE2.
Can you point that out on the left picture there? Kind of like.
So it's like we're looking at one little arm right there.
Oh, okay. One little of those little cauliflowers.
Yep, one side of the cauliflower.
Oh, I got it.
Oh, I guess this guy might be easier to point it out on. Right there. Yeah. Okay, so now we're going to talk about where you can go to learn more about COVID-19 related proteins, or really any protein your heart desires that there is a solved structure for. So the little background before I throw it over to Catherine. Catherine's going to show you a data bank with a whole bunch of protein structures in it. All of these protein structures have been solved experimentally by scientists called structural biologists, and they spend a lot of time growing crystals of proteins and then shooting them with X-rays and using the diffraction pattern of those X-rays to inform a model like Catherine will show you and like we saw with the spike in ACE2. There's recently, in the last 10 years, a new technology that's come online called cryo-electron microscopy, where you can just have a bunch of proteins and then you freeze them, get them really cold, and look at them with an electron microscope. So you shoot electrons at this surface that's coated with these frozen proteins and look and see how those electrons bounce off. And that bouncing off a computer can turn into a really nice micrograph. And you can see then not just, so in a crystal, you have proteins frozen in time in ways that we can get them to crystallize. But in cryo-EM, we can see a variety of different conformations. And so it can inform some ideas of how the proteins move and flop around in a biological system. So over to Catherine.
All righty.
Let me pause my screen sharing.
Okay, so the site that's going to be most useful, first you're going to want to just Google PDB, and you're going to want to go to the RCSB PDB. PDB stands for Protein Data Bank. And you should come up to a page that looks like this, and you can type in whatever protein you may desire to look at. So for our purposes, let's look at SARS-CoV-2. And you'll see a bunch of different structures pop up. How you'll want to choose a structure is generally look at what method was used to make it. We generally like X-ray, I think diffraction and crystallography, but we want to avoid NMR. I don't see any that are NMR here. And generally you want a angstrom, a resolution that is less than 3, preferably less than 2.5. The structure that we have been liking a lot for analyzing COVID and ACE2 is the structure with code 6M0J. So this structure, Allison had it earlier in her presentation. It has a portion of the ACE2 receptor and the SARS-CoV-2 spike RBD, or receptor binding domain. And they're already bound together in the crystal. So that's really helpful because scientists have already determined for us how they interact, and they've just given us basically a picture of that interaction that we can then examine closer if we want to. So if you want to really get into the nitty-gritty of proteins, you're gonna want to go to UCSF's Chimera and download this software. Just Google Chimera.
So Catherine's just showing you where to go. This will all be on YouTube. Don't feel like you need to follow along step by step right now. We realize that downloading a program while on Zoom is not the best for everyone's Wi-Fi.
No.
It took me a very long time to download Chimera, but I also have very slow Wi-Fi, so hopefully it's faster for you. But just scroll down here and pick whichever one works for your computer. And when you get it downloaded, new share. Can you guys see the blue?
Yeah.
Yes, we can.
Once you have it downloaded and open it, actually when you first download it, it won't give you this exact scene. It'll be, I think, just a gray box or a black box. But don't worry, you're doing it all right. You're gonna go to File, either at the top toolbar or in the dialog. Either one is fine. And you're gonna click Fetch by ID, and you're gonna type in the ID of whatever protein you wanna look at. So for ours, if you remember, it's 6M0J. And it's just gonna be the title of the protein that you found on the PDB. Let's go back here. Just type in 6M0J and press fetch. And if you're connected to Wi-Fi, it'll just grab it from the PDB. And when you're using Chimera, you're going to want to have a mouse with a scroll ability, because when you interact with the molecule, in order to zoom in and out, you're going to have to scroll with your mouse. To rotate it, you're going to left click over the molecule. And to translate it in the XY plane, you're going to press down on your scroll middle button thing, and you can wiggle it around. So other useful tools that will help with visualizing, you can go to presets.
Welcome to the Visual Energy Measure diagnostic video. I think you'll find.
Doc, are causing problems.
Sorry.
You're going to go to presets interactive 1. And that will color, if you have a model with multiple proteins it'll color the different proteins different colors. If you only have one protein in that model, it'll color it with rainbow coils, which can be a little distracting, but if you have 2 proteins in one view, it's very helpful to use that preset. So here we can see this is the coronavirus RBD, spike protein RBD, and this is the portion of the ACE2 receptor. So other things that might be useful in your exploration, if you hover your mouse over any part of the protein, it'll tell you which residue you're on. So it'll give you the 3-letter code for that amino acid. In this case, it's THR. I'm not sure what that is. And then it'll also give you which number it is. So that's the 20th residue on this protein chain. And it will also tell you what chain you're on. So this is chain A. So every single protein in this chain in the blue, it'll have an A at the end of its name because it's telling you that it's in chain A. You can figure out which chains you have by going to select chain, and it'll pop up with the letters of the chains. And you can see chain E is our spike protein and chain A is our ACE2 receptor. Other useful things, you can color specific amino acids by pressing Control and left click. You can press Control Shift click to select multiple amino acids at the same time. And then you can go to Actions, Color, and label them a different color if you so desire. Other useful things that you might want to look at, you can go to Actions, Atoms and Bonds, Sidechain / base, Show. And that'll show those R groups that Allison was talking about earlier. So if you want to examine the interactions of these two proteins more closely, you can show all the side chains that are along this interface on, say, the N-terminal helix here, and also this loop of random coil. And just look to see if you see any areas that are where the atoms are particularly close together, if you notice any atoms that you think could potentially H-bond, or other interesting interactions. That's all stuff you can do in your free time for fun and for personal enrichment. And yes.
Do you think that you had a knack for 3D visualization, or that and that if a person didn't have this like ability to interpret 3Ds on a flat screen, they would have a hard time with this stuff? Or let's see. Because it looks
it's hard for me to say because I've always been a very visual person, and I've never existed as a non-visual person, so I can't really tell you if it would be harder or easier. But even for me, I don't know, having grown up with technology a lot and also being a very like see things person, like learning through visual stuff and 3D models, it was still a bit of a learning curve to just more master all of the commands and stuff in Chimera. So don't get disheartened if you find it frustrating at first. We all do. But it's really quite rewarding once you start fiddling around with it. Yeah.
Because, you know, when you're flipping it, I know some people would have a difficult time saying, okay, I just flipped that. Where the heck did that other thing go?
That's why you can color things different colors. That way,
yeah, that's a good trick.
You can always know where it is.
Yeah.
Yeah. And I have a techie question, because I have no idea this, but you know, like a long time ago, I said, well, what DNA is a right-hand helix, right? So why?
It has to do with the chirality of the molecules.
Yeah, I know, but why the chirality? Like, why did nature choose that?
Nature seems to like right-hand rotating things.
I mean, collagen's a left-handed helix.
Yeah, that's one of the very few. Yeah. I was just curious if you kind of knew that. And there aren't many left-hand helix in proteins, right?
Right.
Because collagen is the only one I ever knew. And I just kind of like stumbled across that once. I said, wow, how come left-handed? And it's not.
I think you can get some polyprolines that can be left-handed.
Okay. Just curious.
Yeah, so that's Chimera. Other stuff we do in the lab that you can poke into on your own time is we like to use the autodock vena function, which allows you to dock small ligands to a protein receptor. And there are a lot of very good YouTube tutorials online of that you can just find in your spare time. And another very useful one is the Matchmaker tool, which just helps you visually align proteins that are quite similar. So 2 proteins, one from like a cat and one from a human, it'll sort of mush them together and show you which parts are very similar and line them up. Yeah.
Can you explain an example of a project in the lab that's using the Matchmaker tool?
Oh, yeah.
Thank you.
I think, let me see, which one's the most fun? I think Traces is pretty fun. I think he's lining up the human ACE2 with a camel's ACE2 because it was found in a paper that, well, found in a paper. A paper stated that the SARS, I think it's CoV-2, or was it a different virus?
MERS.
Okay, MERS cannot bind to the camel ACE2, but it can bind to the human ACE2.
Right.
Or SARS. That's SARS.
SARS. Okay.
Yeah.
It can bind to the human one, but not the camel one. And he's trying to figure out why exactly by lining them up and seeing which parts are different, and also doing comparisons of their primary structure with the sequence alignment. And
get thirsty.
Camels get thirsty too.
Yeah. And could you also explain why looking at something like how a small ligand binds to a protein might be important for how we're figuring out how to treat SARS-CoV-2 in the clinic?
Yeah, so most drugs that we use, a lot of them are all small ligands. They're very small molecules, and it can be very handy to see, just as like a preliminary test for eliminating candidates, using a computer to quickly dock them and see if they're even feasible to block a specific pathway or a receptor or act as a competitive inhibitor. Yeah.
Yeah, and that's a great segue into the rest of our talk. Thank you, Catherine, for the Chimera and protein database tutorial.
And it just goes to show you that if you're a minor genius in life, you can actually learn Chimera in 3 weeks.
Yeah. You too can learn Chimera in 3 weeks if you put your mind to it.
And watch a lot of YouTube.
And watch a lot of YouTube.
So that's where you can learn more about all of the proteins that play a role in COVID-19 and SARS-CoV-2, but also any protein that you're interested in. You could look up insulin or hemoglobin and just really get a good picture of what that is and what it looks like. So now we're going to talk about how you can help at home with no biochemistry expertise beyond this talk. So distributed computing through a program called Folding at Home. Just Google Folding at Home, it'll take you right there. So distributed computing is a technique where instead of building a giant server farm and linking all those servers together to make a supercomputer, you can, over the internet, link together a bunch of computers and then use that computing power from that internet linkage. So it uses the computing power of your computer when you're not using it. So you can set it to only do it when your computer's in standby, so it won't affect your Netflix streaming or your Zoom calls. But when you're outside in your garden and your computer's just on, then your computing power can be used to help solve the pandemic. So Folding at Home is a really, really cool distributed computing program. And so they've just published an open access paper. So that means anybody can read it. I encourage you to, about how they've been working along with all of the citizen scientists that have contributed their computing power to model protein folding, and also because they've done all the structural analysis they cared to do, and we have some really good structures now. They're now moving on to what Catherine was talking about, small molecule drug discovery. So looking at all the different places molecule used in a drug can bind to help either, I don't know, destroy the virus or prevent it binding with your ACE2 receptor, or break the RNA polymerase inside the virus. So all sorts of different things. And that's really computationally expensive. And so that means that it takes a lot of time for computers to do that work. And it just takes a lot of computing power, lots of little gigabytes and widgets doing their things in the computer, solving all the math equations. So we can see here in their figure from their paper, the number of people who have donated computer time to Folding at Home over the course of the pandemic. So number of Folding at Home users are in blue, and then the number of COVID-19 cases is in orange. So you can see that we've just at the end of May, maybe mid-June, reached the number where Folding at Home users contributing computer power reached the number of COVID-19 cases. And so you can see different milestones along the pandemic so far.
It's interesting too that just the interest in folding home grew enormously, probably because of the COVID-19.
Yeah, probably.
Yes, very quickly.
I want to live.
Yeah.
How can I donate my computer instead of my blood?
Yeah. So they do also work on other diseases. So they have HIV projects.
Yeah.
They have some hereditary disease projects. So I have not personally signed up my computer for this yet, but I think you might even be able to pick what project your computing time goes to. So if there's some other disease that they're working on that you really have a heart for, you can sign up for that instead.
Yeah.
And just as a shout out for the Claremont Makerspace too, when they went into shutdown, they actually dedicated their spare computing power at the Claremont Makerspace to Folding at Home.
Wow.
And encourage their users. You can join teams. So apparently the Claremont Makerspace has a team that you can join in on in part of being part of Fold at Home.
So before I forget this, Catherine made a video of how to do this folding with Miriam and maybe someone else, I think, too. And I was looking it up on the internet just a moment ago when you might have heard the wrong thing come on. And I couldn't find it on YouTube. Can you guys recommend what that site is too?
It's on our YouTube.
It's on our YouTube. Is it a public YouTube as well?
I thought we had made it public.
Okay.
Catherine, do you remember?
It should be public, I think.
Right after this talk, I will go and confirm that it is public, in which case you can find us on YouTube at NH Sci. And there should be
like Vanna White.
Yeah. we should have now or in the near future a video with Catherine and 2 other students explaining a lot of the same protein modeling, how to get started.
Yeah, good.
Yeah, the Folding at Home stuff is new though.
Okay.
Yes.
Yeah. Oops. So you can see.
And so what do they type in for if you're in YouTube? They type in New Hampshire Academy of Science or something.
New Hampshire Academy of Science. Yes.
Okay. All right.
And
I'll look for it.
Smash that subscribe button. It's called Guide to Protein Modeling Tools.
Okay.
And I will ensure that it is public.
Oh, so that's how it works. We can put some things public on our YouTube channel and some things not.
Yes.
I get it. Well, yeah, this was really interesting.
We're not done yet.
Yeah, not done yet.
So you can see this map of where everyone has signed up for Fold at Home, and every little dot is one user.
Yeah, the whole country of Japan is on Folding at Home.
Yeah.
Yeah.
See us right in there. There's probably a big one for Claremont Makerspace.
Yeah.
And what's really cool is they've built a supercomputer with the processing speed of exaflops.
Yeah.
So giga is times 10 to the 9th. Tera is times 10 to the 12th. Exa is times 10 to the 18th.
Yeah, that's ridiculous.
And the
Yeah.
first exaflop scale supercomputer wasn't predicted to come online until 2021. So this distributed computing network has really outpowered every supercomputer out there. You can see some really specialized, really important research group ones. We have the IBM Summit right there. We have Sierra over there. So these giant server farms strung together to make a supercomputer. And folding at home has just really surpassed them.
Wow.
Scientists.
The power of the pipples.
Yeah. So all these citizen scientists contributing has allowed for really speedy and really high level computations to be done.
Yeah. So did Leon Snow, did she help? I can see Leon is on.
Yeah, she's in the video. It was me,
Yeah.
Miriam, and Leanne.
So it's you, Miriam, and Leon. Leon, when you were doing that, when you were doing the 3D modeling, what was the biggest challenge that you faced?
I don't think that's Arlie.
Maybe she's muted and she doesn't
Oh.
know.
That's to you, Catherine. Or also she was on shaky Wi-Fi's.
Oh, right. Poor Leon. She
Wait,
looked-
I thought he asked Leanne.
Oh,
Leon, Snow.
I don't think she's on. I think it's a
No.
different Leanne.
Okay. All right. Well.
oh, it's a different Leanne who maybe she has also done protein modeling. Was not part of our summer program.
All right. Just random protein modelers tuning in.
Yes.
Well, this is good. You recorded this too, right?
Yes.
Good. This is a good presentation. I want to see this up on the public part of our website.
It's also on it will be on the Claremont Makerspace YouTube.
Oh, good.
Yep.
Good.
So I'd just like to acknowledge the New Hampshire Academy of Science. We do work with the Fairbanks Museum and Planetarium STEM Lab. We run a lot of their science research there. And we, of course, collaborated with the Claremont Makerspace, which is how a lot of you found us. And we're supported in our outreach by National Institutes of Health SEPA grant.